and Houston. It gives me very nice pleasure to introduce and welcome Marty Pagel. Good morning, Marty, and I hope things are good in Houston, where you are at the MD Anderson Cancer Center. And I guess if the cancer community comes with the message, Houston, we have a problem, then you have the answer, because your title is Molecular Imaging of the Tumor Microenvironment. Marty, if you're online.
Just want to start by thanking you and the other organizers, and also Dr. Sylvia Aime for the invitation to join you all here today. I've always been inspired by Dr. Aime's work, and quite literally 20 years ago was the first time I learned about his CEST MRI research. It inspired me to leave industry and move to academia to pursue CEST MRI as well. So I owe him a lot of credit for my career, and I want to thank him very sincerely. Just a few conflicts of interest. I work with a number of companies, have a number of collaborators and funding sources. I'd like to focus on the tumor microenvironment, particularly pH, vascular perfusion, and oxygenation. And with regard to pH, I think many of us are familiar with the Warburg effect, where glucose is processed into lactic acid, which then gets secreted from a tumor cell, or there can be compensatory mechanisms that neutralize the lactic acid inside the cell, which causes extracellular acidification. In either case, the extracellular microenvironment can be below a pH of 7.0, and that's been shown with a number of techniques. Here is an example of CEST MRI data showing a controlled tumor that has then been upregulated to have high carbonic anhydrase 9 or high LDHA. And with both of those overexpressions, you do get a lower pH of these tumors that is statistically significant. Another example of measuring pH, which is somewhat timely, is I've been getting some inquiries about measuring tumor pH relative to infection. Infectious diseases tend to be pH neutral or even alkaline, because they can produce bacterial and fungal infections can produce urea and ammonia and become a little alkaline. That can be seen here with an infectious model with valley fever, showing neutral or slightly high pH relative to normal, and tumors again are very acidic. So it's very easy to tell these two conditions apart. And of course, with residual disease from COVID-19 that will affect patients for years, now understanding if the benign yet inflammatory nodules that are seen on a CT from COVID-19, if they're truly benign or if they're cancerous nodules, and therefore a tumor is, I think, another opportunity for using pH imaging. Well, how do we go about this pH imaging? Well, to describe this chemical exchange saturation transfer effect, or CEST effect, I have this experiment here where this molecule, iopamidol, a clinically approved CT agent that Dr. Aime first started to study with a variety of techniques, including CEST. Well, we've put this molecule inside the small tube, and in the outer tube then is just pure water for reference. And if I were then to selectively saturate a frequency of 15 ppm on the MRI scale, well, nothing on this molecule has that frequency. Water is defined to have a frequency of 0 ppm. So the water signal in the inner tube and outer tube is 100%. But now if I were to selectively iterate a series of frequencies, and you can see different images here as I selectively saturate different frequencies, eventually I do saturate at 5.6 ppm, the frequency of a proton on or hydrogen atom on this molecule that exchanges with water. And this saturation or scrambling effect to remove the coherent magnetization from this hydrogen atom, which is then transferred to the water, causes water signal to go down. You can see the inner tube is looking a little darker than the control outer tube. And I'm going to then hit the arrow key, and you may notice that this tube gets lighter and darker. So if you focus on that tube, you can see it gets a little lighter and a little darker, and there is a second effect now appearing at 4.2 ppm, from the other protons on this molecule. And then, of course, if we directly saturate all of the water in the inner tube and outer tube, that becomes very dark. And for fitting purposes, we often take a symmetrical spectrum in this particular field. And there are a variety of ways of analyzing the data, which I'll get to in a moment. But one way is just to fit with Lorentzian lines. And you can see that there are two peaks. There are a variety of paramagnetic agents that Dr. Ayame and others over the years have studied. There are some diamagnetic agents, such as these aryl amides that I'm showing here. And then a variety of endogenous materials that can be imaged with CEST MRI as well. So how are we measuring pH? Well, this molecule with iopamidol does have these hydrogen atoms that are exchangeable. A hydroxide ion can diffuse past the molecule, grab that hydrogen, new hydrogen atom takes its place. And importantly, there are two types of hydrogen atoms that I'm highlighting in red and blue, and they exchange relatively slow and get faster as the pH increases, or they start fast and get even faster over time as the pH increases. And CEST is really tuned for a certain chemical exchange rate regime. So if you go too fast, you actually lose that CEST effect. And that can be seen here where this suppression of the water signal actually disappears as the pH gets higher and higher for one of these protons, but not for the other. It actually gets lower. So I can take a ratio, a rather complicated ratio, log base 10 ratio. pH is a log base 10 measurement, and identify or calculate the pH. Now, one of the themes that I really want to focus on here is the importance of image analysis. And I know many of us are very familiar with that importance, but I think it really can't be understated and really is good to emphasize. So that's my focus, my take-home message throughout this presentation. And here is one example. The easiest way of analyzing these spectra is to look at the asymmetrical properties on one side relative to the other. And so if I were to compare both sides, you can see that there is some asymmetry, and those are the so-called CEST peaks in this spectrum. This is for a paramagnetic agent where these peaks can be resolved fairly well. There's also the Lorentzian line fitting method that can be easily accomplished to find these peaks, find these dips, and then turn that upside down to find the peaks in the spectrum. We actually use the fundamental equations of how magnetism evolves during MRI studies and model the data and fit that to the experimental data. And that handles noise quite well. Fitting with block equations works better than Lorentzian line shapes for noisy data. So again, it seems obvious to many of us here today, but thinking of how you're going to analyze the data does affect how you acquire the data. And I find very often in our small animal imaging facility that those who have less experience rush in, acquire data, don't think about the analysis very thoughtfully. And then after spending a lot of money and time to acquire data, then they start to think of how to analyze it. They realize that mistakes have been made. So it's good to think of how you're going to analyze the data, which can then affect your acquisition. Here's another example of that. This happens to be a clinical CEST MRI study of the endogenous proteins within triple negative breast cancer. So not using that contrast agent. And this is really telling us about the amount of protein material, small proteins within the tumor. And I can saturate with a large amount of power or a smaller amount of power. And I can do fittings with this asymmetry analysis or Lorentzian line shape analysis method. So which is best? How should we analyze or acquire and analyze the data in the most synergistic way? Well, as you can see with high power, the spectral features are very broad, and yet the signal to noise is pretty good in this spectrum. So you can see some asymmetry properties quite nicely, while at lower power, there's more noise, so it's more difficult to analyze. The Lorentzian line shape method, though, is more difficult to analyze at high power because the peak is very broad. The more narrow peak caused by a low power saturation is easier to model with the Lorentzian line fitting method. So Lorentzian line fitting is better at low power. This asymmetry analysis is better at high power, as you can see. And we found that with a study with about 25 or 30 breast cancer patients, somewhere around there, the low power method with Lorentzian line fitting analysis showed the most significant results as the patients were being treated with neoadjuvant chemotherapy. And so we can see that the amount of proteinaceous material in the tumor is decreasing over time as an indicator of early response to therapy. So unfortunately, high power using that same method did not work as well. So again, you really have to think of how you're acquiring and analyzing data together when you're doing biomedical imaging, obviously. So I'd like to switch to a different imaging method known as multispectral optoacoustic tomography, or MSOT. This is also known as photo-acoustic imaging, or PAI. I tend to use the MSOT term because I work with a company, iThera Medical, that likes to use that term. So that's just my favorite term, but either term is fine. This works by using a near-infrared laser, which is then shined onto the mouse or the tissue in the clinic as well. And that will cause certain molecules to absorb that light and then undergo thermoelastic expansion if these laser pulses are pulsed at the right frequency. Now, this isn't a chemical vibration of bonds. It's more of a macroscopic effect of the entire material breathing and expanding and contracting in a synergistic way, which can then produce an ultrasound signal, which can be recorded. And well, as you know, ultrasound can image very deep into tissues, and we can actually get red light or near infrared light deep into tissues as well. The problem with optical imaging is getting the light back out in a coherent way, which is why most optical techniques are only a few millimeters deep into the body. But if we can blast the tissue with a lot of red light, near infrared light, and get it very deep into the tissue, we don't care if how the light is coming back out. We can detect the ultrasound. Therefore, we have about a 3-centimeter depth of view. The instrument that we use, the MSOT instrument, places a mouse in a bag material so that it is submerged in a water tank, and then passes through this 270-degree detector. So we get very good sensitivity with this machine. We also have a clinical instrument. The only requirement for the patient and operators is to wear some laser safety goggles just for safety, although it's a relatively low power laser being used. So it looks like a rather large, bulky ultrasound device. So very easy to use. But here's a key. MSOT is really not an anatomical imaging modality. MRI, CT produce much finer images for looking at anatomy. And here's an example where in the mouse, you can kind of see the kidneys, intestines, and so on. But you have to somewhat be trained to feel that you're looking at the correct tissues within the MSOT image. Where this modality really has its strength is in molecular imaging, because the spectra of oxyhemoglobin, deoxyhemoglobin, melanin, perhaps also indocyanine green and other dyes, can then be spectrally unmixed from a series of images that are acquired at different frequencies. And this can then isolate the signals of these different molecules. So this is a molecular imaging technique looking at oxy and deoxyhemoglobin, perhaps melanin, and then an exogenous dye, and not to look at the anatomy. So what can we do? Well, here's an example of dynamic contrast enhancement MSOT, where we're actually able to quantitatively measure vascular perfusion in these tumor models. And here's a tumor along with a blood vessel, also known as an arterial input function. We can find this blood vessel extremely easily with MSOT because the machine is very good at detecting hemoglobin. Unlike MRI, where it's challenging to find the blood vessels, or CT, MSOT has no difficulty finding the blood vessels. So we can find the signal from that arterial input function and the tumor. Now, if we can convert to concentration, then we can use standard pharmacokinetic analysis equations and make maps of the wash-in rate and the washout rate. But here's the problem. We can't actually convert this data to concentration. And this gets back to the theme here. Now you really have to think of how you're going to do image analysis while you're acquiring the data. The analysis is really, I think, a bottleneck in our field. So the problem is that the amount of light that gets into tissue is highly variable, and there could be a lot of light at the surface of tissue, more light deeper into tissue, and so on. And because of that, you can't really say how much light got to the molecules of interest, and therefore you can't convert to a molecular concentration. So we've avoided that issue by normalizing the data to 100% for both the blood vessel and the tumor, and then reworking our equations to really use a normalized type of a set of equations that really looks at the change in signal, delta sigma, between the tumor and the blood vessels. And so it's actually a cumulative average, which helps with signal to noise. It's a linear equation when we finally work it out. And so that works out quite well for our analyses. We can then measure this normalized K-trans wash-in rate or the KEP washout rate. Just some quick data to show that we do take out this effect of the variable fluence, the variable amount of light that gets into tissue. You can see there is a gradient if we don't account for that. If we do take that into consideration and use the correct equations, we can avoid a gradient effect within the tumor. Here's an example where I'll really just focus on the right here of a normoxic, mildly hypoxic, and very hypoxic tumor model that has high, medium, and low vascular perfusion. And then after radiation therapy, we can see a strong decrease in the normoxic model, mild decrease in the hypoxic, mildly hypoxic mild, and an increase in the hypoxic model, showing only temporary damage to the blood vessels, but not enough to cause a cure for the cancer. This is a very easy method in our laboratory now. It's all publicly available. And so I really think there's opportunities that DCE MSAT can replace DCE MRI for a lot of small animal imaging. I think the precision is better than in MRI. Now, another reason we like this photoacoustic imaging modality is that we can have a mouse breathe in medical grade air at 21% O2. We then collect a series of images, do spectral unmixing to get the oxy and deoxyhemoglobin maps, and then we can calculate those maps for 21% O2, or we can go back and also have the mouse breathe in 100% oxygen and get that map of the percent oxygen saturation as well. So then we could take the difference between those two maps to calculate what we call a delta SO2 map. Now that can be difficult to interpret. My interpretation is as follows. I think of it as the available oxygen capacity within the tumor. Now, similar to if you're driving in a car and you notice that your tank is a quarter tank full, well, it's also a 3/4 tank empty. So it has an available gas capacity of 3/4 of a tank. Well, in this way, we can see that the maximum amount of oxygen that we can get into the tumor is our maximum tank. So we can calculate how much available capacity is in the tumor to take up more oxygen. And hypoxic tumors just can't take up more oxygen, as you can imagine. And that can be seen here. I won't dwell on this data because it is a bit complex to see all these maps and the subtraction and so on. But we do get data showing this delta SO2 and the percent SO2s with the two different gas carriers. What I'd like to focus on is this, because this really tells the story. So I've taken these percent SO2s and delta SO2 values and plotted them here for you for this normoxic, mildly hypoxic, and hypoxic tumor model again. If we just focus on pre-treatment oxygenation, you can clearly see which tumors are normoxic, mildly hypoxic, and hypoxic using either one of the gases or this available oxygen capacity. Again, hypoxic tumors just don't have any capacity to take up more oxygen. But after radiation treatment, there really isn't a clear pattern here. So this is not a post-treatment biomarker. It can only predict which tumors are normoxic or hypoxic and should respond to radiation. Afterwards, it's less clear. But what we did notice is that this change in the available oxygen capacity, this delta SO2, decreases remarkably for tumors that do experience good effect from the radiation, the normoxic model, while the hypoxic model doesn't change at all. So again, there's really no effect, no meaningful effect from this radiation therapy on the hypoxic model. And that could lead to this chart below, where if you see a certain amount of change in delta SO2, you would know how to change the treatment for the patient. And we're starting to translate the oxygenation studies to the clinic right now, as you can imagine. So just real quickly, again, to emphasize this importance of image analysis and how you're going to manipulate the data later on. This is sort of a paradigm in our lab and somewhat in our small animal imaging facility here at MD Anderson Cancer Center, where we like to develop a study plan for each project. We circulate to everyone, make sure that everyone agrees with the plan before starting. And then we review this every few weeks to make sure that everything is up to date. And importantly, that includes data transfer and archival, the plans for the analysis, image processing, which I consider relatively routine, like drawing a region of interest on an image, maybe curating the data a little bit, and then more sophisticated image analysis, which could include such as a MATLAB calculation program. And then finally, your deliverables, the write-up of the results, I think is part of the analysis as well. And to further emphasize that those 4 parts should really be part of every study plan that you put together for biomedical imaging, in my opinion. Well, here is the imaging pipeline that we often do in our laboratory, where we're making molecular imaging agents. On occasion, we test them in vitro. Not very often. We like to go directly to in vivo and acquire data. But you can see that the second half of this entire pipeline is analysis. So the acquisition, perhaps not drawn to scale, is a significant part, but so is the analysis. And again, I think it's often underlooked and underappreciated. So these are the 4 parts again. And finally, I'd like to make a case that for each hour of imaging, you should really plan on 2 to 3 hours for image analysis. When setting your budget for a project, that can often be overlooked. Scanner time, the number of mice, personnel who are acquiring the data, that's all calculated. And then it's easy to forget that it takes somebody's time to analyze the data, and it can take a lot of time. And often that is not considered appropriately within the budget for the project. So I'm perhaps over emphasizing this. We all understand the importance of analysis, but I do think that more attention really has to be given to these 4 separate parts. Finally, I'd just like to thank others in the lab who I work with. They've been great at putting some crazy ideas into practice, collaborators here at MD Anderson and elsewhere. And finally,